now that we've gone through how to decode the genetic code, we're going to look at how proteins are actually built. And the process of building proteins is called protein synthesis, and it takes place in 2 steps. The first step is called transcription, and the second step is called translation. And a way to remember that transcription happens first is that transcription is first alphabetically because of the C in it compared to the L in translation. So transcription is where a strand of DNA is copied and it makes a strand of RNA, specifically mRNA. The location, or where this takes place, is in the nucleus. And what happens is an enzyme binds to the DNA and it signals the DNA to unwind. So you can see that in this area, the DNA is not twisted up anymore. After the DNA unwinds, the enzyme can read the bases in one of the DNA strands. And so in this particular strand, it looks like the bottom strand is being read. So this enzyme is this RNA polymerase. And its job is to read the bases in one of the DNA strands. So it reads this and sees, oh, here's an A. And it calls out to the cytoplasm to send in a complementary base to A. And so it brings in a U. Remember, RNA does not have a T. It only has a U replaces it. So then when the RNA polymerase reads a T, the RNA polymerase calls out to the cytoplasm for an A, and the A comes in. And the new nucleotides, the ones in red, are making the mRNA, will bond together and make a strand of RNA. Then, once that happens and that mRNA strand finishes, the mRNA moves to the cytoplasm, and then translation will take place. Translation happens on a ribosome, and so the molecule of mRNA will leave the nucleus and move to the ribosome. The ribosome will consist of 2 pieces of rRNA, there's 2 pieces here, and other proteins, and it reads the sequence of codons in mRNA. This steps 2 and 3 are like the decoding steps that we practiced in the last video. Then molecules of tRNA will bring the amino acids to the ribosomes. These tRNA molecules are just floating in the cytoplasm. So when they receive the signal to head to the ribosome, they do, and they bring their adjoining amino acids. Then once they're at the ribosome, the amino acids get joined together to form a chain of amino acids, and this chain will keep growing until they reach a stop codon. Then that chain is released from the ribosome, and then it will fold, the chain will fold and be processed into its final protein structure. So here's a little image of it. Sorry, it's kind of blurry. And so here, this blue area is the nucleus. And you can see that there's DNA in here. And that DNA is going through transcription. See this unwound area, which makes the strand of mRNA. And that mRNA is able to leave the nucleus. And then once it's in the cytoplasm, which is this whole yellow area, it joins up with a ribosome. And remember that ribosome is made up of 2 pieces of rRNA. So here's the little piece, the small, and the big. Then we see that the tRNA has the amino acid on top, and it comes into the ribosome where it attaches and the amino acids join together to create this amino acid chain. And once the stop codon is reached, then this chain will release into the cytoplasm. And so that is how we make proteins.